I'll I'll request Professor Ashish to give his introduction of Professor Anuj.
Welcome, learned professors, researchers, and dear students. COVID-19 is knocking our doors. More than 15,000 persons get infected in last 24 hours in India, which is third highest after Brazil and America. So it is necessary to help our nation to fight against corona. So we have here, first of all, Dr. Anuj Mubayi. Anuj Mubayi is an applied and computational mathematical scientist whose research is driven by the mathematical modeling of problems of interest to the public health or social science community. He has extensive experience in the health decision analysis and in successful development of a strong mathematical sciences training program. He is an associate director at Precisioner and held faculty position at Simon A. Levin Mathematical Computational Modeling Science Center at the Arizona State University, Temple. He also co-directed Mathematical Theoretical Biology Institute and was earlier Associate Research Scientist at the Prevention Research Center, Berkeley. He is a research member of the National Alliance for Doctoral Studies in the Mathematical Sciences and the Intercollegiate Biomathematics Alliance. So I request Dr. Anuj Mubayi to start his talk, and I request all participants to give their questions on questions and answer blog. So they can post their questions, and Professor Prasad will inform Dr. Anuj Mubayi about their questions, and he will answer your questions. So thank you. So Dr. Anuj Mubayi.
Anuj, can you share your presentation, Anuj?
Yeah. So I will switch off my video because just to have the better quality of the presentation. So can you see my this thing?
Yes. Yes. I'm sending live.
Okay.
Yeah, it's online now.
Okay. Thank you.
You can go full screen. Yes. Yeah, we are able to see.
Okay. So greetings, everyone. Good morning, Tuesday. Good morning in India, and it is evening here, late evening here in US. It's my pleasure and honor to speak. I was invited by Dr. Prasad, and it is always, I know him for a long time, and this was, I mean, we chatted on various things in the past, and it's really pleasure and as well as honor to be part of this COVID-19 modeling seminar that he and his collaborators have organized. With that, I would like to first say, give my gratitude to Dean Mary Saril for telling me so many things about VIT. I was not really aware of. It seems like an amazing institute. I may have to apply for a position there at some point of time. And also gratitude to Professor Rishi Kumar as well as Ashish Kumar. But they are all organizing is not an easy job for especially an event like this one where so many people remotely are being joining to this this webinar. So with that, let me give what I would be talking about today. Before I go into the details of it, I want to be very clear that I'm not an epidemiologist. I'm not a clinical expert. I'm only a mathematical modeler and help understand disease better and study control and evaluations of the control program. So such as social distancing, basically. So my training has been, my PhD is in applied mathematics, almost more than 10 years ago. I had an experience working in Department of Infectious Disease in a hospital. I also was part of a clinical trials before. And currently I'm part of organization called as Precision Here, where I'm associate director. And we work on health economics problems and evaluating control programs and cost-effective ways, suggesting some of those policies, basically. So with that, I will go further. I was thinking what would be the best strategy to talk today. I just 2 days ago gave another webinar, and so kind of out of ideas, but eventually pulled out together some of the things that is very relevant to COVID-19, but also give you understanding about how, as a modeler, we think. And it is very important that we focus on our systems and model it accordingly. So first, I will talk about some historical challenges, background. I will also talk about epidemiological aspects in COVID-19. And then I will talk about some of my studies on modeling COVID-19 aspects, eventually ending it with future modeling challenges and limitations. So, the pandemics have nothing been new to us. Maybe in my lifespan, there has been now. Prasad, can you hear me properly, or is it from time to time my connection is not very good?
We can hear you, sir.
Okay, fantastic. There has been many pandemics in the past, and we have learned quite a lot by analyzing data and using models. What has been successful and what has not been successful in the past. For example, in 1918 Spanish flu, we did not know even about the virus, that it is spread by influenza virus. It was spreading by influenza virus. It is usually people used to think it is maybe a bacterial infection. It was not until 1933 that the virus, human influenza A virus, was isolated in humans. So it was very late. One of the key features that recently was published almost 2 decades ago, or less than 2 decades, was published about using data from 2 different cities, which was one was Philadelphia and the other one was St. Louis. You see on the top right of my slide, there is a 2 graph of Philadelphia and St. Louis. Philadelphia has a very high peak, and St. Louis had a very low peak and spread out outbreak. And one of the reasons that the people that analyzed the data for these 2 cities recognized that the reason that Philadelphia has a high and a bad outbreak in some sense, because they waited 2 weeks after their first case to implement the social distancing policies, whereas St. Louis implemented it in 2 days. So that resulted in something we have been hearing quite a lot in the news. It's called as flattening the curve. And the flattening the curve is on the right. You are seeing if you implement certain control measures, then basically the peak comes down and the curves kind of flatten up. And well, but there is an issue here that we can't just implement the social distancing policy and flatten the curve, because there is something called as a hospital capacity. Because we don't want to overwhelm our hospital capacity, and that is a horizontal line. And you see the 2 horizontal dotted lines are there. One is the initial before an outbreak, and slowly you want to raise that health capacity of the hospitals so that it can handle more cases. For example, in New York, they had this big area conference venue that they converted into hospitals. So they increased the number of beds for the patients of COVID. So this line is actually not stable. This is fluctuating line. So there's uncertainty associated with it. And this keep on changing daily, on a daily basis, and beds are getting added. there is a lot of variation in these type of things from city to city. Well, the flattening of curve has now you don't have to, if you have left your health capacity to a previous to COVID level, you may have to do a lot more social distancing protective measures. Then slowly when you increment your health capacity, you may relax social distancing and maybe open up the cities, for examples. So these are these variations. Usually modelers, as a modelers, we have to think about not only in terms of a disease outbreak, how many cases, what is the peak and all those things, but also as a level of healthcare capacity that could handle number of cases that are occurring for the COVID outbreak. So this is the timeline for the diseases, for the COVID outbreak, starting from early January. In fact, in China, it was in December, and some says even in November, there was a first case reported. We started to see the big news about COVID-19 starting January. And well, as a modeler, what is important is that uncertainty in the information has changed. For example, initially, we only know a little bit about epidemics characteristics, whether it is spreading in clusters or whether it is spreading everywhere, or whether there is asymptomatic cases are spreading it, and so on. So this information slowly and slowly started to become more widespread, and therefore the uncertainty in the epidemic information was reducing. But then there was other information that was coming as far as policies are concerned over a period of time that we started to see uncertainty in. So the evolution of information that we use in the models have changed drastically from being more less precise to more precise now than before. And there are still many things we don't know about. And if we model things using those information, we may be limited by our capacity of modeling methods to draw a reasonable conclusion. So this slide is about evolution of models and evolution of information that is used in the mathematical models, for example. Well, I came across this BBC interesting article, actually interesting in some sense, but it's sad in other sense. And it was on, and I kind of thought over this thing quite a lot. This was written by a journalist. She's an Indian, and one of her family member died in Gujarat fighting with COVID. And well, she writes about how devastating things have been because our system is overwhelmed, because there is a fear in the systems. And these things are very critical for the models to understand, because human behavior plays a very important role in the spread of the diseases. So what she says in this article is, so as the infection was, I mean, as her relative was, his condition was getting bad, she was doing many things, and she was a BBC journalist, so she's very well known. In fact, she knew many people, in fact, many of the political leaders and other journalists, local journalists in India. So she said, I called hospital after hospital. I was told there was no room, not a single bed in the entire city. Repeatedly calling helpline, got nowhere. Finally, after being admitted one day of his ICU, intensive care unit, I was able to see him struggling for oxygen in a video call with me. A strong man who was exercised daily and had no health issues. He could never have thought that a virus would land him in this situation. Well, it is really painful reading this article, but really an eye-opener that there are hospital settings or healthcare system and it's not about India, it's about whole world I'm talking about. They are being overwhelmed. Usually in developing countries like India, they have been overcrowded, understaffed, long wait hours because resources are limited. It's a developing countries, not a developed country. Even in developed country like US, in New York, in spite of being the best city, has to face many of these challenges. So there was no ventilators, for example, there was no good communication. So now the question comes, what as a modelers we can do for that? I mean, of course, we read these stories every day and we become sad and we kind of post these things on our social media accounts. But what exactly we as our strength is a mathematical modeler. So what can we do basically? So, well, there is lots of models that are already established by mathematical modelers in the past. We can use many of those things. So for example, queuing theory based models have been used for management of hospitals, for example. So if you see top left corner, there's a picture where there is circles and circles donate the hospital settings. So this person who is as a patient is coming in to some centralized hotline and asking about where are the beds available, you need to we need to streamline it effectively that they don't die on waiting to get into line for getting a bed, for examples. And as happened in this case in BBC article, for example. So the queuing theory has been amazingly helpful. The models using queuing theory here, we talk, there is certain inputs that goes in such type of model and it optimizes where an individual should go, which hospital individual should go and where is the availability, rather than randomly going to the hospitals and wasting time and basically making risk for the life of a patient, basically. So just to give you an idea, this model has input like arrival process. For example, how many patients arrive in a queue in a given hour? What is an average time interval between arrivals? Does that vary? The arrival of the people coming to the hospital depends on a time of the day, whether it is morning, lot of patient coming of the COVID, whether evening is it. Queuing process, how much time patient is waiting. Suppose you are in the hospital, that is a best availability that you can get. How much time you have to wait there to get the bed finally. So these kind of service, for example, what would be if the hospital is overcrowded? How do you decide first come first? How do you decide who will get the first bed, for example? So there are lots of these inputs that goes in the models, and it helps you answer certain output of the model that I have it in my bottom right. For example, it helps you optimize the results over average weight per patients. How can you reduce average weight patients? What percent of time that a healthcare employee or a machine that is used on a patient are being utilized? You don't want to waste the time just by sitting around where other places person is dying because there is no machine, for example, or there is no people to healthcare worker peoples. Here also you can talk about capacity, raw materials like PPE, protective gears, for example, or ventilators. How many you need in a certain location? So these things can be done very interesting using a queuing theory based models. And behind the scene, they are using something called as a little law, which describes the relationship between number of patients in a system and a mean arrival rate. And the length of patients remains in the system. So how much duration the patient stay in the hospital, for example. So very beautiful modeling techniques have been used in the past but it seems like in India we are not using it, and in other countries also these modeling techniques we are not using, that we can use it, for example. Okay. Well, another story that I came across in a scroll.in, it's an Indian story that talks about there was in BHU, there's a diagnostic lab. There's a Institute of Medical Sciences. That's a Department of Institute of Medical Sciences in Banaras Hindu University. And there is a diagnostic lab that is handling 30 million people, and for just one test lab is testing for 30 million people. Think about that. That's a huge number of people. It's one lab in Eastern UP, Uttar Pradesh. So it seems like we are understaffed. The testing facilities are very limited, and well, in this case, they are getting samples every day, 1,000 samples they can handle in 48 hours. They take 48 hours also, and they are getting around 1,700 samples a day. So, well, huge amount of samples they are getting. So they are prone to make an error into it. Because they are overwhelmed with the work and they are dealing with a lot more people, a lot more region. These tests will be prone to lot of error. So how do you model those errors, for example, and what those errors will lead to? How many new cases? What changes it will do to the outbreaks? So I mean, there's a models for imperfect testing on the disease dynamics. You can talk about imperfect testing. Now, in this picture that you are seeing here, there is one patient that we are following, and these colored lines indicate different types of test. Well, there is antibodies test, there is antigen test, there is actual virus detection test. So it depends on what you are testing. So PCR is a virus detection test. Polymerase chain reaction is the test that basically identify a live virus in the body, in the specimen or sample. The only time you can use it if the live virus is a lot in the body, but as the progression or initially you may not find the actual live virus because it is very small amount of virus in the body, and the test will say no, whereas the test may make a mistake there in identifying. Or later on also, after a person has recovered, if you do the testing using PCR, you may not be able to find the virus, and it will give an error. It will say the virus is not there, whereas the virus is there, but at a very low level, right? Then there is another test called as antibody test, where antibodies are getting. Antibody test you cannot find in the initial stage of the infection because antibodies takes little time to develop in the body. So this test starts to fail if you do it very early on. Antibody test will not give a correct result. So the question is, how do you model? And how, if you model it, how do you understand the disease dynamics on it, basically? So, well, before I go into the actual model component of imperfect testing on the disease dynamics using these two tests, PCR test and antibody test, we need to understand what is the main, how do we say that the test is creating a bad result, for example. So for example, so if you see this table is showing test being positive and test being negative. If a person is infected, if the person is not infected on the columns. So there were total infected, their total number of people in the population was N. Out of N, A plus C were infected and B plus D were not infected. Okay, so A plus C plus B plus D is equal to N. That's the total population. Okay, so the question here is, if you apply a test, you can fail to detect or you identify it correctly. So there are 4 different choices. The test is saying positive and the person is positive. That means person is infected. Or the test is positive, but person is not infected. Or test is negative. That means test is saying not infected, but actually the person is infected. Test is saying not infective, but the person is also not infected. So in 4 of these values, 2 times you made an error and 2 times you made a basically correct decision. So test sensitivity, any test has something called as test sensitivity and test specificity. Basically, it captures how many test sensitivity captures how many correctly you have identified among all the people who are infected. Okay, and test specificity is how many correctly you have identified among those without the disease. Okay, so this is A and D and divided by total population in that column, by the way. So these are the two parameters that people use for identifying how bad is the test or how good is the test, basically. Okay, so there is simple model we can use for it, is SIR model, which is susceptible, infected, recovered, which is a simple epidemic. And here you can build in virus active test, in infection test. So there are two possible tests: active virus infection test, which is a PCR test, and then an antibody test, which is basically testing for antibody. And here the testing rate is basically given by sigma A and tau A for active virus, and antibodies you are testing it by sigma B and sigma tau B. And basically it depends on antibodies test. You will not do it in a current infection. You will do it in a quarantine people who are already recovered from the infection. There you want to see if you have an antibodies or not. Whereas active infection test, you will do it in a general population because you don't know if they have the, so you will do the in unquarantined individuals. By the way, QS, QI, QR are quarantined individuals here. And you can build in sensitivity and specificity in the simple models, and you can study what happens if the error made by the test varies. And error could vary because of many different reasons. Because of the test, the raw materials of the test are bad, or test handles in a very incorrect way. So these things we can study using a very simple model like this one, and study what happens over time if the test efficacy we know is this much. Okay, and that was happening in the BHU, Banaras Hindu University Diagnostic Test Lab. They were making errors, but we don't know how much of that error is causing the outbreak to grow or reduce, and over what period of time. And such kind of models can be used very easily to understand this thing. Now, again, I'm giving you a broader perspective about the modelings of various relevant features in the community with the proper examples that we are seeing currently, rather than just making up something here. Okay, so this is a very good example and very related to the examples that I gave from the Banaras Hindu University Diagnostic Lab. Now, well, another aspects in the modeling that we face is about definitions. Well, I think some of you might be aware of, but this is something came in May 22 news that almost 40%, 35%, 40% cases are being don't have a symptoms, and if they don't have a symptoms, how will you know that they are spreading the case? So they are. What is the name we should use for that? Whether we should use latent, whether you use asymptomatic, whether you use pre-symptomatic. What is the name? And each of these words has a precise definition in the literature. Okay, so for example, asymptomatic means people who are not showing symptom, but will never show the symptoms in future also. They will recover without showing symptoms. In previous diseases, we might use this kind of terminology in a different sense, but here asymptomatic is used like a self-healing, where you don't see the symptom at all. And pre-symptomatic are the individuals who are right now asymptomatic, but eventually will going to show the symptoms. And these two folks, these two groups are causing lots of infection in the communities, especially in the COVID case. So definitions have to be properly modeled in our mathematical modeling equation before actually we talk about mathematical analysis, numerical simulation, linking with the data, and so on and so forth. So this is one thing, and I think if there will be lots of cases which will not be showing symptoms and spreading the disease, then the best thing you can do is either not go out at all out of your home, social distancing, or wear the face mask. So one could even evaluate the impact of these measures more accurately if you define the terminology in a proper way. So we'll talk about some modeling methods now, and some of you might not have a background in modeling methods for pandemics. So let's talk about for an outbreak. There are two basic questions that we talk about: contagiousness, that means how fast the disease spreading in the population, and you might have heard about the number which is called reproduction number. That is very common in the news articles nowadays. And then the second one is how virulent or how dangerous is the disease. That means how many people dying from the disease. And these are captured by case fatality ratio or mortality rate of some sort. In this table, you see there are different diseases with different reproduction number. Measles has a high reproduction number, but not medium case fatality ratio, whereas MERS had a very high case fatality ratio, but had a very low reproduction number. Low reproduction number, how it is spreading, and case fatality ratio is how deadly it is, the disease, basically. Okay, well, this case fatality ratio is why I'm talking about these two metrics. Let me go back actually, why I'm talking about these things. Well, this is relevant when we talk about a disease like COVID, these two outcomes of your models. If we do not provide these outcomes from your models, then we are giving only partial information about an outbreak. These are critical informations. And besides that, you can do many other things. But I think some of these aspects needs to be incorporated very much in your as an output variables from your models, basically. So case fatality is basically a ratio of how many people died divided by how many diagnosed with the disease. Okay, now this is a disease. So how many diagnosed with the disease depends on how many you test, basically. Some countries are testing quite a lot. Some are not testing very lot. For example, South Korea is testing a lot. And so therefore their case fatality ratio will be very low. Whereas in US, India, and many other countries, it is not as high as in South Korea, for example. So that depends on the denominator of the case fatality ratio, which is number of people who are diagnosed. So this metric may not be the best one unless you can do a seroprevalent, no, not seroprevalent, actual sampling study where you can follow the people. And then you can estimate case fatality ratio in the cohort of the samples. So it depends on CFR, depends on surveillance system, testing capability, testing efficacy, but also it depends on who we are testing. So for example, we may not be able to identify asymptomatic and pre-symptomatic cases, which are not showing symptoms. So therefore the denominator will decrease. It could also depend on age. For example, some countries, old people are dying quite a lot, whereas some other countries, some other age groups are more dominant. So, so age structure matters quite a lot when you are calculating case fatality ratio. So this number is kind of delicate and has to be used in a very cautiously. For the output variable. So I'm showing in this picture, right corner, how is the case fatality ratio is changing over time. Why? Because you are testing now more and more. So as the time is progressing, this is in China, in the 4 different places in China: Wuhan, outside of Wuhan, outside of Hubei. Hubei is a province or a state of Wuhan city, and China overall. So this has changed over a period of time because they were doing more and more testing, and therefore denominator is increasing quite a lot. So this numbers is showing inconsistency in the output. But again, it is the best thing that could measure the fatality. Some people also use crude mortality rate. In the crude mortality rate, the denominator is the total population, not the total cases diagnosed, but the total population. And sometimes people use that one to draw a conclusion. But then the number of deaths could be highly underreported also. Well, as a modeler, we ask ourselves many things. As the outbreak is going on, we initially we ask ourselves, how do we prevent? If there are few number of cases, how do we prevent? Like, for example, SARS outbreak, or not SARS, sorry. Yes, SARS also. SARS didn't become a pandemic like COVID. It spread, but we were able to prevent its invasion or reinvasion due to many social distancing measures. SARS outbreak was in 2002, 2003. What is the growth rate of it? How fast it is increasing? The outbreak is increasing. It is very important metrics, and we used many different models for measuring how fast it is increasing. Okay, this is very, very critical, and this could be not necessarily for the whole world or for the whole country, but you can talk about your city or your neighborhood, how fast it is growing. And it depends on the local structure of the community. What interventions would be helpful for the population in hand? How drastic intervention we should implement? These all questions are modeled and are captured and asked by as a modelers. So we do it all the time answering this thing, and we are doing it for COVID-19 as well. Well, before I go into an examples from my research, I need to give some basic background on the this disease models. To some folks may not have an experience in that. So disease model, basically what you do is you consider whatever population that you are trying to study the disease in, you divide that population into three compartments, basic three compartments. Susceptibles, people who are not infected, infectious, people who are infected and infecting others, I compartment, and R, people who have recovered from the disease. And then you have the arrow coming from susceptible to I to R. And that is called as a transmission. That means new cases. Transmission is the new cases that are converting from S to I due to infection transmission, and recovery, that is cases are going from I to R. Now these two arrows are modeled by in a very, very different way. What if people are changing their behavior? So transmission rate will decrease or increase based on how strictly one is following, people are following social distancing, for example. But in general, these two I compartment, if I look, it is a function of something coming in and something going out. And that something coming in and something going out decides the level of I compartments. And you see, if we simulate it, the I compartment will look like a red curve in this picture. Okay. So that's a very simple thing. Now these transmission rate could be a function of host population size, contact rates, how fast they are moving, pathogen infectivity, like how bad is the pathogen in transmission itself. So again, there is a quantity, we call it as a reproduction number, average number of new cases that we talked about a few slides back. It's basically number of new secondary cases each infected person is generating in the population. Now, this is an average number of all, average it out over all the infected people in the population. But this number is beautiful because it tells you few things. It tells you first, it tells you the ratio of how much is coming in and how much is going out in the I compartment. So this is beta over gamma, how much comes in and how much goes out. So if beta over gamma is greater than 1, that means more coming in and less going out, then there will be a peak. That means R0 will be greater than 1 and there will be a peak. If beta is less than gamma, then there won't be a peak. And you see the top two graphs are showing R0 greater than 1 and R0 less than 1. In the first graph, there is an outbreak or there is an epidemic. and in the second graph, there is no epidemic. Okay. So this is an epidemic threshold. That's why we call it as an epidemic threshold, because it tells you whether there is an outbreak or not. It also tells you how quickly you will rise to the peak. So if you see in this graph on the third graph, that higher the value of R0, the quickly it is rising to the peak. There are different values of R0 that this picture shows, and it shows the peak is high and very early on. So it tells you how fast the epidemic is growing also. It also tells you what will be the size of the peak, how many people total will be infected, and it will also tells you what level of immunity or what level of control programs we will do in the population to control the disease. So there are many features of this reproduction numbers that we use in defining an outbreak. Well, just to give you an idea here, these are 4 diseases that I'm comparing: measles, influenza, chickenpox, rubella. And measles has a very high reproduction number, and influenza has moderately high, but chickenpox is much higher than influenza, and rubella is lower than chickenpox but higher than influenza. So you see these graphs that we are seeing on the left side, the peaks, and if these type outbreak of these things will come into population, it will show the peaks like this, assuming everything else the same, basically. Now you can make this type of modeling more complex. For example, recent news that I just heard is that you may not be having a permanent immunity of the COVID. You may recover a small period of time, and then you become susceptible to disease. You can get the disease, and in that case, it is not simply SEI model, or E stands for hidden infection, asymptomatic, let's say. But the second graph would be more appropriate if you are talking about that you may get the infection again. In that case, the recovered compartment would come back to the susceptible compartment. Okay. Prasad, can you let me know 10 minutes if I'm before my end period of 10 minutes? Yeah.
Okay, Anuj, I'll do that.
Sure.
I think you are now till 20 minutes is there.
Okay. Thank you. Thank you.
Yeah.
So in this models, you can make it very complex. For example, you can incorporate T compartment, which is a treatment compartment. Although for COVID, there is no treatment, but the treatment also means providing ventilators, for example. Treatment doesn't mean only drugs here. So treatment is therapy, some kind of therapy that you are providing. So how do we model? Which models we should use? Of course, it depends on a research question in hand, but it also varies upon what shape we are trying to imitate. So if you see on the bottom right corner, it is showing a different shape based on what you incorporate in the model. So what we are seeing right now in the COVID is a third situation, which is we are expecting the second wave in some countries, by the way. In India, it is still a first phase, but second countries, there are countries which are expecting the second phase right now. So it could be the last figure that would be a better to model that way rather than first 2 figures, for example. Well, as a modeler, what do we do? I mean, I think some of us do these things so regularly, but we never discuss. We never discuss about it. There is lots of uncertainty in our task, basically. So precisely what we do is these 5 things as a modelers. First is identifying the sources of the mechanism. That means we have to know the research questions. We have to know what are the factors that are driving the research questions, and that may be critical to the dynamics. So 30% of our work efforts goes in the just identifying those sources. And then incorporating those sources in the mathematical models, 10% roughly. That's very small amount as compared to identifying the source of mechanisms. Well, performing the mathematical analysis and statistics is also, these are not absolute numbers. These are variable number, but approximately 20% of the effort goes in mathematical because we are used to doing these mathematical analysis. And then interpreting the results. Interpreting the results takes a huge deals of information and connecting it back to the first point, which is mechanisms. And that takes 50% of the, and eventually we need to validate the results using an alternative source of who says that our results are correct. You have to find a ways to identify how do you validate these things. So that these process we do it now and then, but the amount of percentage, according to me, it should be like this distribution. The 30% in the top one and the 25% in the bottom one are the most important time that we are spending in understanding our mathematical models. There are different reproduction numbers that we talk about in the literature. There is lots of uncertain aspects. There is confusions because people use left and right reproduction numbers, but the meaning of it is very, very different. So the basic reproduction numbers that I talked about is only at the starting of an epidemic or starting of an outbreak. Effective reproduction numbers is a time dependent and you can talk about any time. Control reproduction number only do you talk about when the interventions or the controls or some kind of like face mask, social distancing is already there in the population. Then you talk about control reproduction numbers and type reproduction numbers when you might have a different type of people, right? For examples, you could have age group. So you can talk about old type reproduction numbers versus young type. So how many old people are infecting and how many young people are infecting, for example. So that is called as a type reproduction number. Now invasion reproduction number is also there. That is when there is multiple strains. So suppose COVID pathogen strain mutate and there are maybe 2-3 strains of it circulates in the population. In that case, you will model those multiple strains. In dengue, for example, there are 4 strains. And people model it. There you talk about when one strain is there, what is the reproduction number of other strains to invade? So there are many of these reproduction numbers that there are other reproduction numbers, but these are important ones. And so I listed it here. And so there should not be a confusion. We should always ask her, people use reproduction number, but which one you are talking about? And the meaning will change and the measurement of that will change as well. Now, lately we have been hearing something like a herd immunity, and one of the examples of the COVID comes into mind is Sweden. Sweden is actually doing something called as they are trying to achieve a herd immunity, and that's why they didn't do any kind of lockdown, and they let people do everything except they said, okay, you have to be careful, don't be too close. It was really in informal ways that they are kind of pressing forward to that. Well, a recent study. So if there is a question.
No, yes. You can continue. And there are 10 more minutes are left for you.
Okay. A study showed 6.6% Sweden population has antibodies only till late May. And actually 60% is assumed to be herd immunity. Herd immunity means that you should have sufficient number of immune people in the population that infected person, when they go out, they don't get enough of people, susceptible people around them to infect others. And so some people are providing immunity to other people, and that is what is called as a herd immunity. So it is believed that 60% people have to be immune in order to be herd immunity. Well, it is calculated in a very, very simple way. It is calculated using R effective reproduction number. And if the effective reproduction number is less than 1, that means you are not growing, the cases are not growing. Each case is not replacing him or herself. basically. So it is R effective or effective reproduction number is R naught, basic reproduction number, times the ratio, how many people are susceptible, fraction of people who are susceptible. And if this number is less than 1, then you find out proportion of susceptibles that needs to be, if you make this thing less than 1, you can compute something called as herd immunity. Very simple idea. And we are talking very fancy way, but it is computed in a very, very simple way, how herd immunity in this thing. So anyway, and as soon as the herd immunity goes up, our effective number goes up, there is a second wave, there is a risk of second wave coming back, basically. So let me talk about some contact tracing modeling. The question is, we don't have a treatment, we don't have a vaccination. So what can we do? Well, contact tracing is one of the best methods. But there is a meaning for it. The people use word quarantining when they are basically isolation is another opposite of quarantine. Both way you are restricted some way. Quarantine is more voluntary, whereas isolation is forceful, that you are asked to remain inside. And basically what you do is, if you are infected, you basically, if they identify the person is infected, they trace the contacts. So how do you model these things? Basically, you can model something like that. If there is a model like SEIR, and basically forced is an isolation, Q3 is an isolation, and Q1 and Q2 is quarantine, because you don't know in Q1 and Q2 whether they have a symptoms or not. They don't, from their face, they don't have a symptoms, whereas I is showing the symptoms, and therefore you are isolating it forcefully. Okay, so so Q1, Q2 is quarantine, and Q3 is isolation here. And you can model this thing very beautifully, contact tracing, even though contact tracing is an individual behavior, but you can model it. So for example, contact tracing, you can say, is fixed efficiency. So if you see my second point bullet here, which is basically capturing how many people from S to Q1 you are taking out and E to Q2 you are taking out, there is K times you are doing more for susceptibles than for exposed class. So that means you are somehow quarantining K people more. So if K is equal to 0, perfect efficiency of tracing. That means everyone who's infected, you are isolating only those, or you are quarantining only the cases which are infected. But people, but if K is equal to 1, K is equal to big number, maybe 50 or 100, then you are basically wasting your efficiency resources. Efficiency is not very good for contact tracing. So beautifully it is done in a very simple way, how the contact tracing could be done. But anyway, I mean, you can talk about cost here. You can model cost associated with it, different types of cost, public health cost, societal cost, direct cost, indirect cost, cost to an individual. We can model all these things very, very cheaply. And there are methods to do that. And this is one of my paper in the past where we model cost associated with quarantining and isolations when the contact tracing efficiency is there, basically. Well, the result is there. What is the tracing efficiency and how much will be the cost? It is not always more contact tracing, always the better. There is a threshold in that, and that would be needed for controlling the disease, which will be cheaper as well as it will control the disease. Well, there is another studies. Prasad, I have 5 minutes, or I'm out of
5
minutes.
5 minutes.
Okay, thank you. I will be done with that. Promise. Well, you know, recently there was a news actually about Delta Airlines changing their policy of boardings. In US, there are planes, they are boarded, people aboard the planes by zone wise, and they take zone 1, zone 2, zone 3, zone 1, zone 2. They have decided it based on optimizing their time. The time is the money for the airlines. So they have done it based on their making so fast so that people can go fast, and therefore we can save money. I mean, the airlines can save money. but that policy may not be true for an outbreak if the outbreak is spreading, for example. So recently Delta changed it. In fact, Delta, American, many airlines changed it from back to front. That means back row comes first, and then the next row from the back, and then the first row in the very end, basically. So the question is, instead of doing the zone wise, which used to be the case, now they are doing is back to front row wise. Is this a good policy, or they just made it a random decision? Well, it turns out, by the way, it spreads in the, it is very much likely to spread in because it is airborne and asymptomatic can spread it, basically. So it is very much through the air travel. So this is the question that we had, whether their policies are better or not in the plane. So we developed these models where basically which we are capturing is probability. These are individuals in the plane. Think about people who are entering the plane and they are susceptible, there are some infective, and they are coming in close contact with each other. And the contacts here defined as the radius of infection as well as duration of the contacts, how long they are next to you, basically. And basically you can model something like that. And we did some of these things with a huge simulations, and we found out the policies that Delta and American Airlines are doing are actually counterintuitive. They are causing more harm than good. In fact, it is better for them to go back to the previous policy. And we just, this paper is actually in the review right now, but it causes lots of uncertainty in that. And this is the way the modeling has been extremely helpful. So with that, let me stop here. Thank you very much for listening to me, and I hope I have motivated enough to provide some basic idea about how we do the modeling, what aspects are critical, how do we relate it to the real life aspects of it rather than just a theoretical aspects of it. So thank you.
Thank you, Anuj. There are few questions from the participants.
Okay.
Yeah, hope you are able to hear me.
Yes, I can, loud and clear.
There is somebody is asking that how China recovered so fast.
How what? Say
how
again.
China recovered so fast.
How China recovered so fast? Well, yeah. It will be too early to say recovered, because China has an outbreak coming. Reporting. I mean, I just read in the newspaper that that there will be this Beijing is infected, is reporting very high number of cases, by the way, and they are closing down all the cities and they are doing the similar type of intervention as Wuhan, by the way. So I don't think that it is recovered, but as I said before, they might have crossed the first wave. The second wave is going on right now in China, by the way. It's starting to.
Okay. There is one more question from one more person. Is saying that some of your models show that there is permanent immunity in COVID-19 case. Is it so?
Yeah, so if you see, my title was Uncertainty and Precision. So, well, we did not, up till recently, we did not know that is it a permanent recovery or is it a temporary recovery? Well, we still don't know very clearly whether temporary recovery is the right way to go forward with the COVID or not. But as far as the past experience on a coronavirus and influenza strain is concerned, for that particular strain, you could, you might have, once you get it, you get permanent immunity. So again, this is what my title was: Uncertainty and Precision. There are so many uncertainty. Maybe in months to come, there will be more studies where we will make a more precise guess, a more precise answer. That okay, there is no permanent immunity, for example, in that, and then we will change our models. But right now, this is the best knowledge we have.
Okay. There is two more questions, and one is some of your models doesn't include SCR type models, only SIR like that only. Why it is so?
Well, the models that I showed were for SIR was just to show, just to build up the theory there. And well, you are absolutely correct that, as I said, the definition of asymptomatic and pre-symptomatic is very, very important. And most of my models right now has been having those type of things, although I didn't showed all the studies, but in the plane also, I wanted to give an idea about how we are modeling it rather than exactly what we are modeling. We do have asymptomatic and pre-symptomatic stages. But again, when we are talking about airplane, for example, the duration is 2 hours. It's not the community that we are talking about. In community, people live for good number of days, basically. But in flight, you stay there for a few hours. So here, the differentiation of asymptomatic, pre-symptomatic really doesn't matter too much. Okay, so it depends on what is my research questions that we could focus our attention to. Yeah.
Okay, this is question from my side.
Sure.
How to validate the models of pandemics such as COVID-19? We have a lot of different data is coming from our side.
Yeah. Well, there are, usually in the field, people use it half the data. If you have 100% data, you use half the data for estimating parameter and another half for validating your work. Okay, so now what will be your half in this case? Well, suppose you have incidence data, like number of new cases that are coming to the hospitals, time series data, that you can use it for estimating parameter. And then you can use a mortality incidence, how many people dying, to estimate, to validate your model, whether that is making sense or not. So usually you have to have some alternative. Well, right now people are also using cell phone data. So how people are moving, how people are following a social distancing, for example. So these apps, we use all these apps, and these apps, once we use it, they report to some collection agency, and we have these data that we are using to identify how people are moving in social distancing, for example. So those type of data also we could use it, right? So there are different data sets that one could use it, one has to use in order to validate. You cannot use the same one, basically, as you are using it for parameter estimation.
I think two more quick questions. Somebody is asking that is it possible to share your PPT?
I think it is a recording you will going to post. Or
Recording I'll post it.
Yeah, so they will have all these things.
Okay. And one more quick question: how to manage the economical aspects with the mathematical modeling during an outbreak?
Well, I showed one example of taking into see when we say economic, what do we mean by that? I mean, economic for who? We cannot do everything. Basically, it's a human mind can understand only limited information. The reality is very complex. And so we need to be focused on, for example, I showed in my slide different types of cost to the individual, cost to the public health, cost to the society. Right? Productivity cost. Productivity is if I am sick, I don't go to the work. That is a cost. There is a money dollars amount that I am not earning, for example. So that is a cost. So what we are measuring it, first of all. And again, the same way that I showed, I mean, you can find out average 1% cost of doing something and then multiplied using your epidemic model, how many people are showing those type of things, and then you multiply that unit price with the total number of people who are infected. So that will give you a total cost, for example. So yeah, I think Mary Farrell also has a question. I saw her hand raised.
Yes. So thank you. I was just waiting for all the mathematical related questions to be asked in Jen, first of all. And thank you so much for the wonderful presentation. And can you share any case studies in which the mathematical modeling was successful so far in any of the viral infections? Was there any case studies on this? Because we normally don't see the mathematical modeling when we are working on chemical experiments. So can you share with us any success stories of this mathematical modeling and how far it was useful in the past?
Yeah. Well, there are many actually, not one or two. There are many studies that has been successful. Well, in HIV, for example, the whole field has been very successful because of because of mathematical modeling tools, because HIV is a long-term disease and there is hidden, people do not come, social stigma is attached to it and people don't come forward to it. There's many ways, for example, when we run this thing, how many drug resistance cases will be there? So they use these treatment for HIV for pre-exposure prophylaxis, which is like a vaccine for HIV, which is given to the vulnerable population. And well, if you give a lot of treatment in the population, how do you trade off? I mean, the benefit is you are giving more treatment in the population, but then you are causing more drug resistance cases also because you are pressuring the population with lots of treatment. So there is a trade-off. What is that threshold? And we have seen it in some population who has not built a models. Their cases has been huge as compared to some places. And we have done studies in South Africa, for examples. In COVID also. Now, one thing I want to, this is actually a very good question you asked. One thing I want to say here is, if we are talking about projection, okay, how many cases will be tomorrow or day after tomorrow or 10, I think there is not many studies. But if you are trying to understand which group I should, where should I find the clusters of the cases? In Delhi, they are identifying clusters using models. Also because what happens is the case might have reported in this neighborhood, but got infected somewhere else. So this may not be the high risk area. And if you do just by clinical study, you may identify this as a high risk area, but the high risk is this area where the person got infected. Okay, so these things, identification of the right clusters, in Delhi, they are doing something like that using mathematical models, for examples. And I think other areas also there people have done it in how the system work is the role of a mathematical, not really as a projection. Not necessarily how many cases will be there or how many deaths will be there. That is weaker things to project because there's so many uncertainty. But you can understand what drives the system, what aspects of the models are changing the system's behavior, basically. That you can understand it. Yeah.
Okay, thank you so much, sir. I think this is a more informative presentation by you. And I would like to address the question what you have asked me in the beginning. And I think first time, I mean, we are in contact and you are in VIT, I mean, virtually. And I would like to extend my invitation to you to come to the campus and you can visit us and you can join with the speakers, I mean, the organizers here who is known to you. So we would like to have collaborations with you and maybe it can be a joint proposals or joint publications and also you can be the visiting professors. So we will be very much happy to have you. And my invitation is to all the speakers present here. So I'm welcoming all of you to visit campus when the situation is becoming all right. And probably after September or October, I think mathematics department is planning for one big conference, which is in association with Mathematical Society of India. So if everything is getting all right, we may be having you in person. Otherwise, again, looking forward to meet you in virtual mode in the month of December. So thank you all. And once again, I would like to thank the speakers. So
well, let me say one thing actually very quickly. Sorry, Prasad. You know,
it's okay.
I showed an example of this airplane models where the boarding policy, we are suggesting a boarding policy which is very good for it. And that's a very good example of what the question you are asking. That is practically being used right now in decision-making policy, for example. So I think the reason we are making it so that we can quickly come to the VIT conference and give some talks there as well and meet folks. So thank you for your invitation. Thank you.
Thank you, Anuj.
Yeah. Bye.
Yeah.
So I will drop right now because it's too late in the night.
Yes. Thank you, Anuj. Thank you for your time. I know it's very late night for you. Thank you for your invitation. And there are a few more questions. Maybe I'll send you a mail and we can get back to you and I can contact them the answers. I can send them back.
Sure. It's my pleasure. And thank you for inviting me. All of you. It's very, very kind of you all to invite me for this. It's an honor. Thank you.
Yeah. Thank you.
Bye.
Okay. So.